Just about every family is impacted by devastating brain disease. That's why scientists have spent decades looking for cures to ALS, multiple sclerosis, and Alzheimer's. They've identified many promising compounds that improve symptoms, but mostly in mice. Unfortunately, when these drugs are tested in large-scale clinical trials in humans, they often fail. So why is so much time, money, and intellectual energy being spent on trials that don't lead to cures?
The basic framework has been to construct an animal model, typically a mouse, but there are others, that has a match for the human symptoms, and then to use that model to test different drugs to see which one worked.
These mouse models are often made by making a change in a single gene, leading to symptoms that mimic human disease.
It turns out that many different people that have the same diagnosis actually have different mutations. And so it may be that that one original mouse model isn't such a good model for all the other patients that have some other different mutation or mutation in a different gene.
If 95% of patients don't have the same mutation as the main animal model, are we really surprised when that drug doesn't work with most patients in the clinic?
One tool that would help is if we had a vertebrate animal model system that would allow us to make hundreds of new mutations, ideally mutations that match particular patients.
The Talbot lab studies an animal particularly well suited to better understanding the genetic complexity of human disease, the zebrafish.
They survive really well in the lab, they reproduce really well, and have lots of offspring.
The embryos and larvae of zebrafish are transparent. You can look right inside the developing brain, understand how development or nervous system function is disrupted in a mutation.
And we can put them under something like a confocal microscope and image them as they develop. Then we can do things like express fluorescent markers, and then we can track individual cells as they develop and migrate around the brain.
You can even test potential drugs just by adding it to the water and looking for an effect.
On top of all of these experimental advantages, the most important may be our ability to make dozens of different mutants at the pace needed for drug discovery.
To make any sort of genetic manipulation in an animal, you need to take a very, very early embryo that's either only one cell or a couple cells, and you need to do manipulations on that one cell. With a fish, they lay eggs, so that's why it's so easy to do those kind of manipulations on a really big scale.
And that scale, combined with recent breakthroughs in genome editing technology, is opening up exciting possibilities for medicine.
Imagine a patient with neurological symptoms who has a newly discovered mutation in a disease gene. The current model, it would take a lot of effort and a lot of time and a lot of money to make an animal model that particularly matched that patient's specific mutation. So the goal going forward is that we might be able to use zebrafish as a way to make a patient-matched mutation. That'll help us understand the functional consequences of the mutation and might allow new approaches to new therapies.
So after decades of disappointing clinical trials, genetic and information technology may finally be showing a path forward, using dozens or even hundreds of zebrafish models to start deeply understanding the disrupted biology of the brain diseases that impact so many.
I think every family is touched by brain disease in one way or another, and we would like to use the power of the zebrafish to try to make new models and investigate new therapies.